Today I will be talking about marine bioengineering and how it relates to technological advances today. Before we get into the applications of this growing technology, we must first define its purpose and ecological pursuits. What necessitates the use of this technology in marine ecosystems? As recent developments have shown, aquaculture industries, environmental agencies, and marine biomedical researchers in particular have harnessed genetic engineering to address the damage caused by human impact by means of selective breeding and genetic alteration. Scientists might potentially enhance species' resilience to illness and environmental challenges. Let us examine a real example of this innovation. We can observe marine genetic engineering in action through genetically modified salmon. In aquaculture, scientists have used advanced gene editing techniques to enhance growth rates and improve disease resistance in fish populations. A notable case is the Aqua Advantage salmon, the first genetically engineered fish approved for human consumption, which grows significantly faster than conventional salmon while requiring less feed. At first glance, this appears to be a breakthrough in addressing global food security and increasing production efficiency. Putting to use marine genetic engineering, there are potentials and ecological risks. Marine genetic engineering, particularly in the modification of fish species, is an emerging technology with the potential to revolutionize aquaculture, enhance food security, and provide novel biomedical applications. However, these advances come with risks, as genetically modified fish escaping to the wild could disrupt native populations, leading to genetic contamination, reduced biodiversity, and long-term ecological imbalance. So drawing back to the salmon example, it's clear that this innovation is not without its concerns. Even small genetic changes can quite literally ripple through an ecosystem in unexpected ways. For instance, some modified salmon have shown increased aggression and consume more resources than their wild counterparts. So what might happen if these fish were to interact with natural populations? Research shows that if genetically modified salmon were to escape into the wild, they could outcompete wild fish for food and habitat because of their enhanced traits, and potentially interbreed, which might alter the genetic makeup of wild populations and harm their ability to adapt over generations. So as this field advances, balancing the benefits for humanity with these ecological threats will be crucial in shaping responsible policy and regulation. Let's look at some more applications. So while much of traditional avian genetic editing focused on chickens, new research has brought this technology to ducks. Instead of relying on complicated traditional method that needed isolated primordial germ cells, scientists have injected an adenovirus carrying the CRISPR-Cas9 system directly into duck embryos. You may ask, what are primordial germ cells? Essentially, primordial germ cells are the early stem cells in an embryo that give rise to eggs or sperm. In other words, they are the starting point for a bird's genetic line. So traditionally, scientists would isolate and edit these cells in a lab before transplanting them back into the embryos. But the method we're talking about skips that step entirely, which makes the whole process much simpler. This method produced genome-edited ducks without the need for complex cell cultures. So by targeting the myostatin gene in ducks, researchers generated genome-edited lines, opening the door to studying muscle development and other traits in duck. And by the way, myostatin acts like a natural brake on muscle growth, limiting how large muscles can grow to. So when scientists edit this gene, they can alter how much muscle a duck grows, which in effect, tweaking the myostatin gene can lead to a more muscle mass, quite literally explaining why it's a focus in livestock and agricultural research. So why does this method, or practice rather, even matter? By using an adenovirus to deliver CRISPR, scientists avoid the slow, complicated germ cell isolation. This makes genome editing in ducks faster and more efficient, which is critical because ducks are increasingly used in specialized agriculture, whether for meat, eggs, or even ecological pest control. Let's dive deeper and see some visualizations of these methods. This diagram you see here shows the traditional approach to genetic editing in birds, where scientists isolate primordial germ cells, which again are early stem cells that form eggs or sperm, and they edit them in the lab and then transplant them back into embryos. Now this method is labor-intensive and has only been successfully used in chickens. In contrast, the adenovirus method we discussed earlier skips these complex steps, making the genome editing faster and more efficient, especially in ducks. Now this next diagram illustrates the new adenovirus-mediated method for genome editing in birds. So instead of isolating primordial germ cells, scientists inject an adenovirus carrying the CRISPR-Cas9 system directly into the duck's embryo's blastoderm. This approach allows for faster and much simpler genome editing, producing depths with targeted genetic changes like the myostatin gene edits, without the need for traditional complex cell culture steps. As we conclude, let's observe the biochemical significance and ecosystem impact. Ultimately, this technology matters not just for agriculture, but as a tool in biochemistry to decode gene function. So by targeting specific genes like myostatin, we uncover how genetic pathways influence physiology, energy metabolism, and even how birds adapt to their environments. Overall, these insights help us understand how species might respond to environmental changes or adapt over generations, informing both conservation and ecosystem stability. Thank you so much for your time in learning about these technological advancements as in bioengineering. Here are all the resources that I used today.